Greetings to everyone. Welcome to today's webinar, which is the first webinar of Genomic series. My name is Aarti Dara. I'm working as a bioinformatician in Carcinos Healthcare, and I'll be your host for today's webinar. Today, we have with us Dr. Bani Jolly, a distinguished expert in the field of genomics. Dr. Bani will be enlightening us on the revolutionary field of pharmacogenomics in oncology and beyond today. Before we dive into this topic, let me introduce Dr. Bani. Dr. Bani has done her PhD in genome informatics from CSIR Institute of Genomics and Integrative Biology. She has spearheaded numerous groundbreaking projects, including the sequencing of India's inaugural 1,000 whole genomes, and contributed to genomic surveillance efforts, notably in the field of SARS-CoV-2. Where she collaborated closely with the World Health Organization to bolster genomic surveillance capabilities across Southeast Asia. Currently, she is working as a senior scientist at Carcinos, where she is leading the charge in developing and implementing cutting-edge informatics and technology solutions, particularly in the realm of cancer search. Before I hand over the session to Dr. Bani, I would request everyone to kindly mute themselves. And for this webinar, we are going to have Q&As at the end of the webinar. Over to you, Dr. Bani.
Thank you, Dr. Aarti. Thank you so much for the very kind introduction. And good evening, everyone. And I thank you all for taking out time on a weekend to attend this webinar. So a brief introduction about myself. I am Bani. I am a senior scientist working at Carcinos Healthcare in Bangalore in the field of genome informatics. And today I'll be speaking about a very relevant and very important topic in genomics, which is pharmacogenomics in oncology and beyond. Before I move on to the more scientific aspects of it, I would like to point out the myth of the average patient. This image here depicts Pythagoras, whom we have known very well since our childhood times for inventing the Pythagorean theorem. But the very less known fact about Pythagoras is that he had a very large aversion to fava beans. And it is said that Pythagoras actually knew things such as G6PD deficiency existed, which is hemolytic syndrome that is caused following the ingestion of fava beans, which is why Pythagoras actually developed an aversion to it and asked all his followers not to consume them. So G6PD deficiency is very well known in the field of pharmacogenomics and is one of the very initial indications of pharmacogenomics that came into being. I would also like to talk about a very recent case that came in on November 27th in 2023, wherein a doctor was prescribed a very commonly used cancer drug for chemotherapy. And instead of saving his life or prolonging his life, the drug actually caused toxicity in him and he was killed. So Dr. Anil Kapoor was actually diagnosed with colon cancer and the treatment was expected to extend his life expectancy, but he died weeks later due to toxicity to 5-fluorouracil, which is a very commonly prescribed chemotherapy drug. Now, going by such instances that happen very regularly around us, adverse drug reactions. If we look at what is the cost burden of these adverse drug reactions in a global scenario, these increase in costs are due to either increased hospitalization, prolongation of the stay of the patient in the hospital, and additional investigations that need to be done to know what is causing the drug reaction or what is needed to make the patient better. So if we talk about global figures, ADR-related hospitalizations in USA and Canada range from 4 to approximately 30%. In Europe, the number is as high as 2.5 to 10%. And in Australia, the numbers are ranging from 5% to even 18%. And if we talk about the cost that is borne by these countries, so we have data from USA that estimates that annually at least $30 billion are spent for treating patients that are coming in with adverse drug reactions. And in inpatient settings, this can be actually approximately $2,000 per adverse drug reaction. And in ICU settings, it can be as high as even $19,000 per reaction. Now coming to the case of India. In India, there are very scattered studies when it comes to how prevalent ADRs are actually. So compared to rest of the countries, even by all these scattered studies, India still stands very high at 18.2% of instances. And 3.7 to at least 32.7 admissions in India are occurring annually with which are related to adverse drug reactions, which is far beyond what is being reported from other countries. And actually, in India, these cases are very underreported, and many areas actually go unnoticed. So, these numbers are actually just a prevalence estimate, and the real numbers cannot be determined from these. And if we speak specifically about oncology, these numbers can range from as low as 2% to in some cancers like breast cancer to even 90%, where admitted patients are at a very high risk of getting an adverse reaction to the therapeutic regimen that is being administered in them. Speaking of cost, again, there are very scattered reports regarding the what is the cost per day incurred by a hospital in India that is admitting people with adverse drug reactions. And depending on the hospital setting, type of drug that was administered, or the type of treatment that the patient was undergoing, this ranges from as high as $600 per day, which is a report from 2005 from a hospital in Mumbai, to as low as even $5 per day from a hospital in South India. Now, these reports are very variable, and since there is no such large scale study that has been done in India that can determine what is the actual frequency of it, we need to take these numbers with a pinch of salt. So, when we talk about imprecise medicine or the classical medicine approach, there is a large percentage of the patient population for which a particular drug is ineffective. So, speaking about antidepressants, for example, it is estimated that 38% of the patient population which are prescribed these drugs actually are not even getting the benefits of it. And this number is as high as 75% in the realm of oncology. And speaking about a personalized medicine approach, especially in the field of oncology, we now want to shift from this classical one size fits all approach wherein same symptom, same diagnosis, and same treatment is same symptom and same diagnosis seen in a group of people accounts for the same treatment being administered in all of them, which can either be beneficial, it can either lead to no benefit, and it can even lead to adverse effects. And now, since genomics is an upcoming field and is being widely used in clinical settings, we are shifting to a personalized medicine approach wherein the genetic makeup of the patient can ensure that the right intervention is given to the right patient at the right time. And genomes can thus enable this precision medicine shift, wherein if we have personal genomic data coming from a patient coupled with genome scale data and population genome databases coming from different genome sequencing projects, coupled with genomic evidence and analysis and guidelines coming from very well-known consortiums such as the PharmGKB, CPIC and FDA, we can now implement a very affordable pharmacogenetic diagnosis so that before a patient is prescribed a certain drug, we can look for evidence if there is any related to a person's genetic makeup that may cause some problems while administering that drug, to ensure that it is either the patient is getting either a normal dose, reduced dose, increased dose, or an alternative drug depending on what the implication his genome shows. Now, speaking specifically in the field of cancer, so the genomic basis of cancer involves two kinds of mutations. Germline variations are those that are inherited by us through our parents, and these are mutations that occur in our gametes. So, since the embryo has these mutations that have been actually transferred to us through our parents, the entire organism thus carries the mutation, which is in contrast with somatic mutations which occur in the body or anywhere inside the body after conception. And somatic mutations may occur even as early as in the embryonic stage of our conception. And as a result, the patch of the affected area where the somatic mutation occur will always be carried by the affected patient. But none of the gametes will carry the mutation, so these will not be passed on to the further generations. Now, these alterations that can occur in the germline or in the somatic case are either point mutations, wherein one base of a particular gene is changed to something else. So, for example, C here changes to A. It can either be an insertion or a deletion. For example, these three bases here can either get inserted inside the original sequence, or a C here can be deleted, which was originally there. These can be also copy number variations. There are in a certain gene which is supposed to be present in just two of our DNA strands. There are multiple copies of the gene now present, which can also cause certain diseases. And these can be chromosomal rearrangements such as translocations and fusions. So when we talk about sequencing in oncology, we now need to use tumor tissues to detect all these alterations, which involves collecting the tissue, uh, isolating DNA from it, sequencing it, and looking for these particular alterations. And what do these alterations actually tell us? They can help in the diagnosis through telling us about molecular markers of the gene mutations that can help in early detection of cancer. It can let us know about the prognosis of the patient, in which molecular markers can classify cancers and predict clinical outcomes even. And most importantly, it can lead to therapeutic interventions wherein we can use mutations found in a patient's cancer genome as targets of a particular drug therapy. In fact, the field of precision oncology has been expanding very vastly since the past few years, and with a lot of drugs now being approved for particular biomarkers that can be targeted using those drugs. In fact, 82% of the drugs that have been approved for cancer since at least the 1990s are actually targeted therapies, and 34% of them actually require an NGS biomarker to be present to allow that drug to be administered in that particular patient. So targeted therapies in cancer means that we are targeting proteins that are required for cancer growth. And since this is a very precise approach, it leads to better and effective therapy. It reduces any unnecessary and ineffective treatment costs and enhances efficacy, safety, and reduces risk of administering that treatment. And going by these advantages, the US FDA has thus come up with a companion list of diagnostics which encompasses over 30 genes and over 60 therapeutics. So, speaking about cancer pharmacogenomics, so next generation sequencing can now be used to detect somatic variations, which can be predictive of response to a particular therapy, wherein we look for biomarkers that targeted cancer therapy, which are most appropriate for a patient's tumor type. And then we have germline variations also that can be related to drug pharmacokinetics or pharmacodynamics, which are basically genetic variations that tell us the relationship between that variation and what is the response that the patient may have to a particular drug. Now, speaking about somatic variations, genomic profiling of tumors in in a certain set of cancer patients can let us know if they have certain biomarkers such as EGFR, NTRK, or BRAF alterations, which are actually indicative of uh, targeted therapies that can be used, such as tyrosine kinase inhibitors. And other alterations in cancer or other indications in the cancer genome, such as tumor mutation burden and microsatellite instability parameters, can also let us know that if that patient is eligible for immunotherapy with anti-PD-1 or anti-PD-L1 drugs. And if in case no such alteration is found in any particular patient, we can go ahead and administer the standard treatment. So cancer biomarkers are not just therapeutic, they are also diagnostic and prognostic value also. So a variant can be a biomarker that can predict either sensitivity, resistance, or toxicity to a particular drug or therapy. It can also predict what drug can target that particular alteration. It can also serve as an inclusion criteria for clinical trials and also show if that particular biomarker is influencing the prognosis of the disease in that particular patient, or warrants implementing surveillance measures for early cancer detection. Speaking of the therapeutics part, ACMG and AMP guidelines for evidence-based characterization of somatic variants have actually been developed by these two consortiums so that we know what is the clinical significance or the clinical applications of somatic variants that can be used to can be used to monitor a patient's either diagnosis, prognosis, or therapeutics. So the ACMG AMP guidelines categorize somatic variants into a architecture ranging from tier 1 to tier 4, wherein tier 1 variants and tier 2 variants are those that have actual clinical applications, ranging from variants having strong clinical significance and variants of potential clinical significance. So in tier 1 variants, level A evidence variants are those that have a FDA therapy that has been approved for it, or they are variants that have been included in professional guidelines, giving strong indication that these are biomarkers that can be targeted by a particular drug. And similarly, level B evidence tells that there are well-powered studies with a lot of consensus from experts in the oncology field that give evidence that a particular biomarker can be targeted by a particular drug. And similarly, for tier 2 variants or variants that are of potential clinical significance, level C evidence states that there is an FDA approved therapy for that biomarker, but in some other cancer or some other tumor types. And level D evidence similarly states that there are preclinical studies for a particular biomarker or a particular mutation, with a few studies and without consensus. So meaning that they still have a bit of a potential clinical significance, but not as strong as level A or level B variants. So coming to therapeutic biomarkers, these are those that can predict that have to be checked in case we are trying to prescribe a particular targeted therapy for cancer. And if we take the example of breast cancer, so BRAF mutations V600E in all solid tumors, actually not just breast cancer, have been indicated for this combinatory therapy. And speaking of breast cancer, PIK3CA mutations are known as biomarkers for treatment against for treatment with the combination of these two drugs. And similarly, many professional guidelines and so many professional guidelines, such as the National Comprehensive Cancer Care Network or NCCN, they offer indications that how particular biomarkers, if detected, can either be used for indicating targeted therapy for a particular cancer. So, for example, BRCA1 and BRCA2 germline mutations. NCCN guidelines state that if there are no germline mutations present in a particular patient, then systematic chemotherapy may be followed. But if germline mutations have been found, PARP inhibitors can probably be used to treat that patient's cancer. And not just therapeutics, not just looking at what drug to use, these variants in somatic cells or tumor cells of a patient can also predict and also actually establish the fact if there is resistance that may come up to a particular therapy or not. So, for example, KRAS G12C mutation and EGFR D761Y mutation are two such mutations that have a very compelling evidence, clinical evidence that support these biomarkers as indicative of resistance to these therapies. Now, coming to germline, the germline basis of this or germline variations that tell us the relationship between a variation that may be there in a person's genome in response to a particular drug or medication. Now, metabolism of a drug inside the body is a process that includes drug absorption, metabolism, excretion, and thus many genes and many systems of the body come into play. And if anything then goes wrong in the genes that are controlling these processes, then it can lead to some effects on it can lead to some implication of what the drug may have on the body. And now data coming from the Indigen project, which was a project that undertook sequencing of over 1,000 whole genome sequences from India, we came to know that each individual in India carries at least 8 clinically actionable pharmacogenomic variations, which are germline variations now in this case. And certain mutations or certain variations out of these are actually more prevalent in the Indian population as compared to other populations. And clinically actionable haplotypes and HLA alleles, for example, some of them are actually very, very prevalent in India, ranging from a frequency of 2% in the population to even 4%. Now, so this brings us to a very important topic that there are many ethnic differences in the prevalence of these pharmacogenomic variations in different populations, which have been and this data has been summarized in this review article we recently put out. So if we take the example of South Asia, there are certain mutations and there are certain haplotypes in South Asia that are more frequent here as compared to other populations that have an implication of increased risk of severe reactions to certain drugs. So, for example, this commonly used anti-convergent is coupled with HLA-B allele 15-2 has a 10x higher risk of for severe skin reactions, and this haplotype is actually very prevalent in South Asia as compared to other populations. Similarly, dapsone plus G6PD deficiency, which is commonly seen in South Asians, actually is implicated in increased risk of dose-dependent hemolysis and hemolytic anemia. If we speak very specifically about India, anesthetic drugs plus pseudokinase in deficiency can lead to higher incidence of adverse drug reactions in a particular community known as the Arya Vyasa or the yeah sorry Yeah. So this particular combination of a phenotype and that particular drug is seen to be in higher has a higher incidence of adverse drug reactions in this particular community in India. And not just this, mutations in genes such as VKORC1, which is implicated in having adverse drug effects to warfarin, CYP2D6 haplotype, which is implicated in antipsychotic drugs, and ApoE epsilon 4 haplotype which has an implication in the anti-Alzheimer drug lecanemab. So coming back to the story of the doctor who recently died because of 5-fluorouracil toxicity, he actually underwent a pre-screening test to ensure that if he can be administered this drug, which gave him an all clear saying that it was safe to get 5-fluorouracil for him. In spite of that, he got toxicity to that particular drug and died as a result. This is because the variations that are actually indicated right now for toxicity against 5-fluorouracil, the data is coming from either Caucasian or other populations with very limited data from South Asian ancestry individuals and other ancestries. So if we look at only the data coming from the US alone, 10 out of 1,000 patients in the US die from 5-fluorouracil toxicity, which is an effect that is very largely avoidable by genetic screening. So what is like 5-fluorouracil and how is it implicated in such a very important
Bani,
toxicity related effect?
hello. I had a
Yes.
quick question.
Yeah.
Hello. I was just wondering test to detect toxicity to F5FU, is it called DYPD or DPYD, something like that?
DPYD, yeah. DPYD is the gene that is implicated in metabolism for 5-fluorouracil. I'll actually come back to all of this in my next few slides, which will make it more clear about what is the test that is recommended for 5FU. So yeah, pyrimidine drugs are anti-metabolite drugs that are widely used in the treatment of all kinds of solid tumors, including colon cancer, breast cancer, lung cancer, and even head and neck cancer. These are of two types, intravenously administered 5FU or orally given capecitabine and tegopur. So what happens in the body when we are given these drugs is inside the body they are converted into the active form of the drug, whether initially we had the intravenous form or the oral form. So these drugs are converted into 5FU, and only 1.3% of this ingested drug is actually what is used as or what actually is needed uh, for chemotherapy or the target that this drug is looking for, and the rest of it actually has to be eliminated from the body. This elimination process or this portion of the drug of the whole process that causes the drug to be excreted out of the system, in this DPYD gene plays a very important role, and in this entire cycle 80 to 85% of the drug is actually eliminated. So if anything goes wrong in this gene here and the drug is not excreted out completely, or if it is the gene is not able to perform as well as it should, it will lead to an increased concentration of this 5-FU drug inside the body, which can lead to very severe adverse effects, including death. So the DPYD gene, or the dihydropyrimidine dehydrogenase gene, is a gene that lies on the chromosome 1 of our DNA. It encodes the DPD enzyme, which is responsible for metabolizing fluoropyrimidine drugs inside the body. And people with DPYD mutation that causes them to have decreased amount of this DPD enzyme are at an increased risk of severe fluorouracil toxicity. And these patients thus will require lower doses of these drugs as compared to the regular population. So patients with variants in this DPYD gene can have an increased risk of developing severe toxicity or even death from 5-FU chemotherapy, which is why when it comes to DPYD pharmacogenomics testing, the US FDA has added warnings to the drug labels of uh, these drugs against the use against the use in patients with DPYD deficiency. And even ESMO guidelines recommend germline testing for DPYD prior to administering fluoropyrimidines in any cancer patient. And when we speak about Indian data coming from the Indigen project, 3% of people in India are actually at an increased risk of 5-FU toxicity, and 10% of the population who requires this drug may require a reduced dose of DPYD as compared to the standard dose. So coming to not just DPYD, how are for different drugs, what are the guidelines that are being followed or need to be followed for the implementation of pharmacogenomic testing in clinical settings? So clinical PGX guidelines are those that help clinicians understand how these genetic tests should be used to optimize therapy in a particular patient and to ensure efficacy and reduce side effects. And these guidelines all tell a standard system of grading levels for evidence linking these genotype phenotypes or for these variant and drug pairs. And there are a lot of consortiums that tell that inform us of these guidelines, including the Clinical Pharmacogenomics Implementation Consortium or CPIC, the Dutch Pharmacogenomic Working Group DPWG, and so on, including the US FDA that has listed a list of pharmacogenomic biomarkers in their drug labelings. So combining all of these, combining the guidelines given by all these consortiums and groups, there is a database known as PharmGKB that encompasses guidelines coming from all these individual groups. And the PharmGKB database has all the variant drug pairs that are associated with some clinical evidence that indicate that this variation needs to be tested in a particular patient before that drug can be administered in it. So now coming to the levels of evidence that are given by PharmGKD regarding a particular drug and variant pair. These can range from level 1 to level 4, level 1 being the highest level of evidence. So level A and level B are subtypes of the sub-levels of the level 1 variants, and the highest level of evidence for clinical application is actually included in the level 1 variants. So these are variations or variant drug pairs, in fact, with specific prescribing guidelines from either very dependent groups like the FDA, or are supported by at least one publication, or have some strong evidence coming from either studies or clinical guidelines that indicate that this variant drug pair actually has some clinical significance to it. And then level 2, 3, and 4 range from moderate level of evidence regarding that variant drug pair to either unsupported level of evidence. In fact, if we talk about level 1A and 1B variants, there are 98 unique drugs which have been included in the PharmGKB database that have an indication with a variant pair that can be implicated in either drug toxicity or reduced activity of a particular enzyme in the body and so on. Out of these, there are certain drugs that are implicated in oncology or commonly used in oncology that have also been included in these 98 drugs, which are the ones you can see here in pink. So the level 1A and 1B variations and drugs in PharmDKB are actually inclusive of 29 unique genes covering 98 unique drugs and 194 unique variants and haplotypes. And these are the genes that are included in these level 1A and 1B that have a level 1A and 1B level of evidence, out of which 11 drugs are oncology associated that have a variant associated in 7 unique genes, leading to at least 35 unique variants and haplotypes that can predict what is the drug response that the patient may have given the particular variation if it is found in it. And coming to the different variant implications of phenotypes that can be then listed in this database or in any guideline regarding a variant drug pair. This can include adverse drug reaction, and the indication will give if the patient will have an increased risk, decreased risk, or normal, routine, or low risk if a particular variation is found and a particular drug is given to that patient. It can have indications related to dosage modification, wherein higher dose or lower dose will be indicated. Information about drug efficacy, wherein the patient will be labeled as, and where the response will be labeled as either an increased response to no response to unknown response. Also including gene activity, like if a variation is present, what is the level of activity that the gene will have compared to normal? Will it have an increased activity or decreased activity? And thus, what will be the status of metabolism of that particular drug? So the drug metabolism can then now range from based on if a variant or a haplotype is present in a particular patient that is implicated in as a response to a particular drug. He can either be an ultra rapid metabolizer or a poor metabolizer, or the effect may be indeterminate. And this can thus lead to an increased function of the gene or a poor function of the gene depending on the variations present in that particular pharmacogene. So if you look at pharmacogenomics associated single nucleotide variations in cancer, these are actually located in 3 genes, DPYD, EGFR, and NUDT50. So these are the level 1A and 1B indications only. So we can see there are many variations that are linked to a lot of drugs that are commonly prescribed in cancer, including 5-fluorouracil, which can either have effects related to toxicity or efficacy or dosage modification or other effects. And there are also certain haplotype variants or combination of a set of variations in these particular genes that have an effect associated with these commonly used cancer drugs or drugs that are commonly used in cancer patients, which tell us whether the group of variations or the haplotypes will lead to toxicity, change in efficacy, or a change in dosage will be required based on the drug that is being administered. So now that we know what variations to look for in particular patients if pharmacogenomics testing is required, coming from the guidelines from all these professional working groups, we now want to know what are the technologies and methodologies that are available for pharmacogenomics testing. So Sanger sequencing is something that existed when the first pharmacogenomic guideline came out. It is a method involving chemical termination of DNA synthesis, which is highly accurate, but it is a very outdated technology because it has low throughput, is time consuming, and it is very specific to small variants, and large variations such as copy number changes and all cannot be detected through Sanger sequencing. Then the second methodology that can and is being used to do pharmacogenomic testing in clinical settings is using mass spectrometry, which is a method that detects polymorphisms by calculating the mass to charge ratio in a particular sample. Now, even though this technique is highly sensitive, it is very labor intensive, time consuming, and a lot of expertise is required to run a mass spec machine. Then we have genotyping arrays that can be used, in which fluorescent probes for a certain set of SNPs are there on a thing known as a gene chip, and this can be used for SNP detection. and thus it is very high throughput and has a high sensitivity, but it has a disadvantage that since the fluorescent probes are made for only a particular set of SNPs, novel variations cannot be detected through this method. And then we have next generation sequencing, which is a high throughput approach of sequencing a patient's genome, uh, uh, which uh, it- which is done by parallelization of different sequencing reactions. Now, coming to the next generation sequencing part, for pharmacogenomic testing, we can either use a targeted sequencing approach wherein we sequence a selected set of genes, which is a very cost effective approach for testing pharmacogenomic variations, but it limits the scope of testing since we may miss out relevant variants that are existing outside the panel that we are sequencing. And it also leads to a limitation in case of discovering novel genes that can have some pharmacogenomic implications. Then we have whole exome sequencing that can sequence only the exonic regions in a particular patient's genome. This methodology provides a comprehensive analysis of all the protein coding regions of a patient's genome, but in this approach also we miss regulatory variants that may influence drug response. And coming to whole genome sequencing, wherein we sequence the entire genome for the patient, which is the most comprehensive way to do a genomic analysis because it captures all genetic variations, including the non-coding regions and the regulatory regions in a patient's genome. Now, why is so now? If we speak about how genome sequencing has been growing since its inception in the early 2000s, the pace of how genomics is growing is also now bringing down the cost. The cost that is there for sequencing a patient's genome. So in the early 2000, it used to a genome costed at least a few thousand US dollars, but now it can be done in as low as either 200 to $500 per genome. So since the cost of genome sequencing is decreasing as time advances and as no newer technologies are coming up, it is very possible to do genomics at scale in clinical settings, and whole genome sequencing is actually more advantageous compared to other sequencing approaches when it comes to a comprehensive analysis of pharmacogenomics for a particular patient. So the advantages being that it provides a very holistic view of a patient's genome, such that promoter variations in genes such as DPYD, VKORC1, and CYP3A5, which are very important pharmacogenes having level 1A and 1B levels of evidence, variations having level 1A and 1B evidence, these can be covered now through whole genome sequencing. WGS also provides a better picture of copy number variations, which are important for pharmacogenes like CYP2D6 and G6PD. It does an accurate genotyping of UGT1A1 gene and other genes that actually that actually need an haplotype indication to be associated with a particular drug pair. Uh, WGS compared to either a genotype panel or even whole exome sequencing has a better coverage of variant indications that are given by CPIC and other pharmacogenomic working groups, including the fact that WGS provides accurate HLA genotyping, which is also a gene that is implicated in certain level 1A and 1B indicated variant drug pairs. WGS is also a future-proof approach because we can now detect variations that may not be relevant right now or may not be indicated in response to a particular drug. But since pharmacogenomics is a constantly evolving field and the current indications are coming from a certain set of ethnic populations, there may be indications in the future that can potentially link a particular variation to a drug response, and these can now be covered through genome sequencing and can be looked at later. And WGS helps identify complex variants and in genes that such as CYP2D6 and CYP3A5, which have complex structures, and pseudogenes, which require accurate distinction for genotyping. So this is why whole genome sequencing looking forward is an approach that may triumph against all other approaches of current pharmacogenomics testing. providing us a very holistic approach and a very holistic view of a patient's genome, and can tell us a lot of indications can come out of it that can predict a patient's response to a particular drug in the future. So to conclude, pharmacogenomics enables personalized treatment based on individual genetics. It improves treatment outcome by optimizing drug selection, dosage selection, and predicting response to a particular drug. NGS, compared to other methods, allows for comprehensive analysis of of all our genes in a single test and helps identify relevant variations, including those in non-coding regions when we talk about WGS, for a more complete pharmacogenomic profile. And medication responses can vary significantly among ethnicities, and routine NGS-based pharmacogenomic testing can thus help close this gap in the future. So with this, I'll stop. And if there are any questions, I'll be happy to take them now.
Hi. Thank you, Dr. Bani, for such an insightful session on pharmacogenomics and understated. This field is still understated in when it comes to Indian genomics. So thank you for putting light on all the things which are required to bring forth new data into this field. Let's take up questions right from the people, and then we would jump to the questions that we have received through Google Form.
Can I ask a question?
Sure,
Yeah,
go
sure,
ahead.
go ahead.
So great talk, Bani. I was just wondering the unfortunate case you highlighted of the doctor who was administered 5-FU and had a severe reaction to it. So cases like that, would you say the situation in India, is it a cost issue or is it an awareness issue, or is it a mixture of both these factors? That why is there no widespread pharmacogenomic testing for agents that are known to have some sort of adverse reaction linked to germline variants?
Yeah, so in India or in any LMICs, it is a combination of cost issue and awareness issue, but mostly it is an awareness issue, because even though guidelines state that DPYD gene must be tested, must undergo germline testing if a patient is to be administered a 5-FU drug, these guidelines are actually not being followed, not even in India and elsewhere also. Since this was the case of a doctor, the patient underwent the testing. And coming back to the ethnic differences problem also, so after this particular patient was given an all clear for administering 5-FU, after his death, it was realized that he actually had a rare variation in the DPYD gene that may be linked to toxicity related effects. So since we do not have indications for all the variations that may exist in the DPYD gene that can relate to toxicity or have a certain effect on a particular patient, it becomes really hard to confidently tell if that particular ADR will happen in a patient or not. But there are still a set of 40 to 50 variations that have been recommended for testing by a lot of consortiums, a lot of even US FDA, but still it is not being actively followed. So yeah, awareness is definitely an issue. And if more oncologists and more people who are prescribing this drug can access this test more, they will probably start prescribing it more also.
Thank you.
Yeah, thanks, Bunny. So it's a really great, nice talk. So let me take an opportunity to say thanks to you guys, and I think Dr. Isa, Dr. Vinod, Dr. Siv Subbu. You guys are doing tremendous job to find out all the common variants in the local population. So the first thing is I'm so surprised that the DPYD is missed by the screening. It's a straightforward. There are no technical challenges. I think it's a part of Oncomine Plus as well. I think as per the guidelines, they have to screen this. That's very unfortunate. The second thing is that, so do you recommend like gene centric screening or drug centric screening? So for instance, like I'm having like say 10-15 patients with hematological malignancies. So after the hematological malignancies, they go with the cocktail drugs. So it will be very difficult part in one hand. The second thing is that HLA typing. So like there are almost like 12 to 13% of HLA contributing to the pharmacogenomics, right? So with a single test, it's very hard. Like for example, if you want to go for whole genome, it's quite expensive. Again, whole genome, you need to use different pipelines, like different capture kit you have to use. So do you recommend a drug-centric or gene-centric in an Indian setting?
By drug centric, you mean that if a particular drug is to be prescribed, then we test only for that particular drug?
Yes, since like for example, Dr. Vinod and Siv Subbu, they cataloged all the local variants, right? At least they can screen those variants. So particularly now, particularly when it comes to pharmacogenomics, I can say it's a preemptive is better rather than
Right.
as a diagnostic approach. Preemptive is the best option. So, but when it comes to particularly DPYD or, for example, HLE or CYP2D6, like all these things, so if you have a known like variants in the population, that would be straightforward. We can use like it's a single real-time PCR based thing. At least we can do that. Say for example, VCRC. So like say for instance, patient got a stroke, we need to do the warfarin. So it's in 10 minutes, they can be a single SNP based test. The same thing like when it comes to certain drugs, for example, FGFR, rather than going for the whole panel and a lot of, lot of sequencing challenges are there. I think best thing is that if you go on a gene centric or drug centric, there'll be a time saving, like in terms of like to save patient, right?
Time saving, yes, definitely. If we consider that the variations that we know are something that will be the only things that will be present in the population that we want to now, or the patient that we want to now screen. But given the fact that there is very less data coming out of India or any South Asian countries that are telling that this particular variation in this particular gene may lead to toxicity to this particular drug, we still need a very a complete picture of the patient's genome, since these indications may change in the future. So for example, today a patient may be negative for DPYD gene and can be prescribed 5-FU. But this is only based on the fact that we have tested him for the 50 mutations that other consortiums have said that are linked to adverse drug effects. But the
No, I mean,
patient may have a mutation that can be linked to it, but it has not been studied elsewhere yet.
see,
So we
like
will
for
miss out
instance,
on those.
I
Yeah.
mean, in case of like DPYD, say the class 1, class 2, or class 3, for example, like we have 50
Right.
variants in a gene. What if you have a unique variant which is not part of class 1, class 2, class 3? It's going
Right.
to be a novel
Right.
variant, right?
Right.
So in our experience from local population in Middle East, so we have seen that like an average like 12% of novel variants we observe in the local population. So you're right.
Right.
Like sometimes there will be a difficulty with the novel variants. So
Right.
that's the reason I said that like the preemptive, you can consider PGX as a preemptive test rather than like as a post, screening test.
Yes, definitely. PGX is most likely more to be used in a preemptive approach rather than a reactive one.
Okay, thank you.
Yes, Nimani, you can go ahead.
Hello, everyone. Thank you for this interesting session. I have just a short question regarding the technical methods used in PGX. Which is the most effective method? Is it mass spectrometry or NGS, knowing that there is no bioinformatics needed in PGX? Thank you so much.
Okay, so yeah, it actually depends on the amount of expertise that a particular laboratory may have. If the bioinformatics capacity is present, then NGS is definitely the better approach for pharmacogenomic screening, because it will let you know about all the variations that can be looked at from a pharmacogenomics perspective. Mass spec, yes, it will cover any variations that are known to cause ADRs or are linked to toxicity to a particular drug. But if the lab has wet lab capacity and people who can run that mass spec machine, then yes, it is a sensitive approach. It is a good approach, but going by the applications that can come out of using NGS for pharmacogenomics, NGS is any day a better approach than mass spec now, especially in the days to come when the cost of genome sequencing will rapidly fall even more.
Great. Thank you so much.
Great. Now let's take up the questions that we have received through Google form. The first question that we have with us is how the pharmacogenomics can help in cervical cancer.
So, as far as I know, there are not any specific drugs associated, not any particular drugs that are uh, indicated to a particular biomarker in the case of cervical cancer. This is the top level of evidence that I'm talking about, but there are certain drugs that have been linked to biomarkers in all solid tumors. So those are the ones that those alterations or those biomarkers are the ones that can be checked in a patient's tumor genome to see if that drug can be administered for treating cervical cancer. Other than that, there are several clinical trials that are in progress leading to lower levels of evidence for a particular drug against biomarkers in in a cervical cancer related tumor. But since these are trials that are ongoing, the level of evidence right now is pretty low. And maybe in the future, we'll have specific cervical cancer related targetable therapies also.
Great.
I had another question. So the DPYD question that I asked, you
Right.
mentioned briefly that all the variants that could possibly cause the adverse reaction, they are not known. Is that because there is less genomic data known for Indian patients? Is
Not
it
just
because
Indian,
we haven't fully characterized
right, exactly.
all the variants in the DPYD gene for India?
Exactly. So the data that led to these guidelines being formed largely came from Caucasian population. So
Right.
in a global perspective, other populations are generally neglected when it comes to either drug usage guidelines or clinical trials. So there is very limited data coming from countries like India, Africa, other South Asian regions, and so on. So yes, these variations have not yet been characterized, and there are several articles and research publications now that are indicating that there are certain rare mutations in the South Asian populations in DPYD gene that can also be linked to drug toxicity. But there is a lot of work that needs to be done to actually get the clinical evidence for these to be included in guidelines later on.
Thank you.
Yes, Yubi, please go ahead.
Hi, Vani. Very nice talk, very informative. I had two queries. One, when you, like in the case of the doctor whom you mentioned, so are there retrospective studies being done on a larger number of such cases where, the advanced stage of cancer was there, or where the effect of drug was expected to be related to the genome? So maybe like when the person did not respond or died soon after. So are there any studies where the genomes of such patients are studied?
Yes, in this particular case also, the genome of the patient was studied post his death,
Yeah, yeah.
and that is when
Was
it
done.
was found that
In
he
this
did
case,
indeed have a rare mutation.
I got
So
it.
yes, yeah.
In this case, it was done. I'm saying like a retrospective studies on a larger patients population where they may have been such
Okay.
cases. In this case, maybe it was a high profile or he being a doctor, it was noticed. But in cases where there's a sudden death because of chemotherapy or the patient is not able to tolerate, so for research point of view, maybe not for the patient therapeutic point of view, but for research purpose, are there some retrospective studies can be done to perhaps correlate um, the genome of the patient and the reaction it had to the drug?
Right. So I'm not aware if any large scale study has been done in this direction by any group, but there are some scattered retrospective analysis that are done on patients that indeed died of drug toxicity. And in these studies, we can then check that if what was the effect that was seen in the patient and what is the genome now suggesting can be possible, can link it to what is the effect that the mutation present in that particular gene had on the particular drug that was administered.
So
So yes,
can I add something
yeah,
here?
Sure.
Yeah. So for instance, like we are having a large cohort of ADR patients, say 164 patients we are having. We did a whole genome on the patients, and we ended with almost like 140 novel variants. The very unfortunate thing is that the proving pathogenicity of this variant is not pretty easy,
Right.
because
Right.
most of them appear as a benign by in silico properties and in terms of frequencies. Say the DPYD, so whenever you see in homozygous state, you will lose like it's a lethal. Homozygous is lethal. So in
Right.
heterozygous, so we can see it actually, because until unless if you do multiple autopsy, you can find the homozygotes. All these things are heterozygotes. It's not easy to prove. So it will take more time. There's a reason like class 1, and class 2 we have very limited variants. So with time, for sure, like one thing is that a large scale population studies always helps to flag all these variants. That's the only thing we can do in current time.
Right. Coupled with at least some in vitro in vivo studies that actually show that this variant had a detrimental effect on the gene function, which led to the incomplete metabolism of the drug in the body. So that is the evidence that will be required to actually indicate if that particular variation is leading to toxicity related to that particular drug.
Hi, hi, Bunny. So thanks, very informative talk. So India is a very vast country with genetic makeup differing from the south of India to the central India to the east of India and to the northern India. The markers you discussed and the variations in the genes, when we look at US with 3%, in India, is there a geographical variation in terms of these mutations?
Yes, so compared to north and the south population also, we see in the cases we see a very different prevalence in the north as compared to south, even if we consider DPYD mutations. So yes, in India, since the population is very admixed and different populations have actually different genomic markers inside in within their genomes, yes, there is a lot of variability within India also, which is then associated with a different prevalence of a particular variation or different prevalences of different variations in a particular gene that may be related to some pharmacogenomic effect.
Thank you.
Yes, Dr. Jyoti, please go ahead.
Hi, Bunny. Jyoti here. So actually, what I understand now, I can see I'm not going to talk about more of scientific and about the variations. What I want to understand from the point of view of a cancer patient and oncologist, and its use in clinics. So it's like I can see three types of say offerings. One is whole genome sequencing. It's like when somebody does it when he is healthy and he does the whole genome sequencing, he also gets the pharmacogenomics results for himself or herself. That is first
Right.
option. Yeah. And second option is only pharmacogenomics, which can be done at any level, even if the patient is there, or maybe when an individual is healthy. That can be the second offering. Now, the third case, what I feel, what I understood from previous question answers also, that if, because these are, what we're talking about 98 drugs, and for cancer, I think there are 11 genes we are talking about, if I'm not wrong. Is that right, Bunny?
That's it. Yeah.
Right. So for cancer patient, suppose if there is a cancer patient and if he goes to an oncologist and oncologist wants to treat the patient with an optimized treatment using a particular drug, and also he wants to see what kind of dose is going to be suitable for the patient, whether higher dose, a standard dose, or you can say lower dose. So for that, will it not be a good idea to test only for these 11 genes and the drugs associated with that for a cancer patient in place of doing whole genome or whole pharmacogenomics? What will be your approach in that case, Bunny?
So yes, you are right. In the case if the doctor only wants to look at what are the different alterations present in these oncology relevant pharmacogenes,
Yeah,
it definitely makes sense to analyze only that particular portion of genes from out of the complete set.
yeah. Yeah. So I am asking this question because of couple of reasons. One is, of course, time will be saved because we can do 11 genes and we can come to know which variations are present, and then we can give pharmacogenomics report in a shorter turnaround time. That is first thing. Second thing, cost, because we will be doing 11 genes and germline testing. So it will be cost effective for the patient who is anyway going to spend a lot of money on their cancer treatment. And third thing, if we go and tell it to the oncologist that if there is a cancer patient, because germline testing, generally oncologists do not do it for the treatment purposes. So except for BRCA1 and BRCA2, or you can say HRD or HRR. Right. So if I do this thing, if I say that these 11 genes are going to tell me the optimized dose of a relevant drug for a patient, I think that will be useful for the oncologist as well as for the patient. Is my understanding correct, Bunny?
Yes, it is correct, but this is going by only the current evidence that is there in the world right now. So if the
Exactly.
doctor wants
Yeah.
to know that there are 11 genes right now and I need to check
Yeah.
only these 11 genes, then yes, it makes sense to use a pharmacogenomic panel sequencing kind of a thing, wherein we only look at these genes and the mutations that are present in those genes only. But
Okay.
this is a very fast evolving field, and evidence starts coming from the world, anywhere in the world, very quickly based on the pace at which the genomics research is increasing in a particular region. So now
Right.
tomorrow,
Right.
if there is an indication saying that there is another gene that is associated with adverse drug effects related to this particular therapy that is to be administered in a cancer patient,
Right.
we won't have data for that patient, which means that if he wants to now check it again, he will have to take the
Yeah.
sample again, submit it to the lab again, and check for that gene again. But if we did a
Yeah.
more comprehensive sequencing approach in the beginning, we would have already had that data, and only the evidence would now need to be rechecked to see if now whatever is coming from anywhere in the world regarding any particular indication, if that is existing in a patient, and now it can be informed to him also.
Okay, got understood that thing. So basically, I mean, I understand that. I mean, you are right that of course, right now we know about these 11 genes. Maybe there will be certain more genes, or maybe certain more drugs, or maybe certain more variants present, and more adverse effects will be found, and we can get more information. But for a particular patient, if I talk about a patient today, these genes, these 11 genes, and the variants present in those genes for pharmacogenomics are sufficient. Is
Right.
that right?
Going by the current guidelines, yes, it is sufficient.
Okay, fine. Thank you, Bani. Thank you.
Thank you, Bani, for answering all the questions. We have reached to the end of the webinar, and this video will be available online and will be shared. And if in case there are questions which are still left, they can be asked on the Carkino's YouTube channel. Thank you everyone for joining this webinar, and thank you again, Bani, for this insightful presentation. I hope everyone learned new things today, and hope to see you in the next series, in the next webinar also. Hope you have a great day. Thank you.